Uh, welcome, Dr. Jason McDermott. Um, a brief introduction about Dr. Jason. Dr. Jason McDermott is a senior scientist, research scientist, and group lead of computational biology at the Pacific Northwest National Laboratory in Richland, Washington, USA. His background is in microbiology and immunology, and he has worked in computational biology for over 20 years. His current research interests include multi-omics data integration, network biology, and machine learning, with projects in cancer, infectious disease, and soil microbiomes. Over to you, Dr. Jason.
Thank you very much for the introduction. I will attempt to share my screen. Again, thanks for the invitation, and thanks for the nice introduction. As I was introduced, I'm Jason McDermott. I will give a slightly brief introduction on what I do in the next slide, but I'm going to talk about integrating omics to understand complex biological systems and kind of an application in both biomarkers and discovering functional mechanisms in cancer. It's actually where I'm positioned in these talks. It's unfortunate. I have a project that's on the effects of ultrasound on cardiac muscle in culture. And so I've been working on that using similar techniques too, but I'm going to present my cancer stuff today too. So I'm a computational biologist at Pacific Northwest National Laboratory. I have been I trained as in bench biology as a microbiologist, and then as a postdoc, I went into computational biology. And I've worked, now a lot of my work is trying to figure out what to do with large amounts of data that are currently being generated in a lot of biological problems. This is for disease, for things like soil microbiomes, for plants. We have some projects, obviously, in infectious disease and in cancer, which is what I'm going to talk about today. I am going to use a few of my cartoons that I draw from my webcomic, Red Pen, Black Pen. This is not associated with my work at PNNL. So what does a computational biologist do? Well, what I tend to do is ask data hard questions. So this is, trying to find the right questions to ask of this large sets of high throughput data to be able to get insight into the biological mechanisms of disease, and also to identify predictive methods for kind of call them biomarkers, but oftentimes they're signatures of disease that can be used for prediction. So when I say omics data, you've probably heard this term thrown around a lot. What do I mean? So omics data probably originated maybe 20 or so years ago with the advent of the beginnings of high throughput sequencing. Basically, omics data and high throughput technologies have kind of revolutionized certain portions of systems biology research for biology. And these could be things from, as I said, genomics, with sequencing genomes, you can do very quickly and cheaply now. Transcriptomics, when you want to get an idea of which genes are expressed from the genomes. Epigenomics, so that's like methylomics, where you're analyzing the methylation state of DNA. And then what I'm going to talk about or focus on, because Pacific Northwest National Lab has a focus on mass spec assisted omics technologies. And so these are proteomics, where you measure large numbers of proteins, phosphoproteomics, where you're measuring phosphorylated proteins to get an idea of signaling pathways, and then some other kinds of omics, which I'm not going to be talking about today, metabolomics, lipidomics, and glycomics. So these are small molecules, measurement of lipids, and measure of glycosylated proteins or And then there are others. So this is a whole zoo of new technologies that are coming online. So essentially, these all have in common that they can generate large numbers of measurements for different biological components. So these, again, genes, transcripts, proteins, metabolites. And each sample can yield, thousands, hundreds to tens of thousands of measurements. Oftentimes, this is depicted in a heat map that you see at the right. Some of you may be familiar with this kind of visualization, but I'll just walk through it because I think it's useful to kind of demonstrate some of the things that omics can be used for. So this is from a proteomics study that we did several years ago, looking at human tumors from high-grade serous ovarian cancer patients. This is about 200 tumor samples from 200 different patients. These are shown as columns, so along the x-axis, each column is a different sample that corresponds to a different tumor. On the rows are different proteins, and so in this study, we identified about 4,000 proteins that could be observed in every single sample all the way across, and we got measurements for them. And the colors of the cells in this heat map are indicate the relative abundance. These have all been normalized to a single scale, but red is higher abundance and blue is lower abundance. And you can see there are patterns in this data. The patterns come because we cluster the data. So we cluster both on the columns. So we cluster tumors by similar patterns of expression, similar patterns of protein abundance across all these proteins. And that gives you these bars at the top, which are subtypes. And these are molecular subtypes. So these are different varieties of ovarian cancer. And they are interesting because subtypes can be very important in terms of prognosis. So outcomes can be very different if a patient has a particular subtype versus another particular subtype. Some are more aggressive, like the proliferative you see in pink here. Let's see if I can give a laser pointer. Yeah, there we go. So the proliferative that you see here. And some have particular treatments or alternative treatments that might be applicable to them. So in this case, you see things that are, have more of a metabolic signature, and tumors that have a more immune component to them. Likewise, on the y-axis, you can cluster by proteins. And so this is groups of proteins shown in this color bar on the right. And these have been, again, you see the patterns, how these have been clustered together. And these have been clustered based on their relative abundance across all of the different tumors. And the labels that we've put on these are from the predominant pathways that are overrepresented in these clusters of proteins. And so you can see things like cytokine signaling, extracellular matrix interactions, and immune functions for some of these. So this provides an idea of what is driving each of these different subtypes and what kinds of, again, alternative therapies might be useful to try with these patients. This is a retrospective study. So these patients have long since been treated. We had survival data and lots of clinical data, which will come up in just a minute. And so we're not actually developing therapies for these patients, but this is to be used in prospective studies and eventually translated into clinical practice, which I'll talk about in a few slides. So just to give you a brief introduction to mass spec based proteomics. This is an older slide, but it really does show the elements of what's happening. Some of the underlying technologies, actually there's multiple underlying technologies, and they're coming out with new mass spec machines and approaches basically on a yearly basis. And as I said, PNNL is kind of a center for this in terms of development of approaches and also implementations of state-of-the-art methods. So on the left, you start out with a complex mixture of proteins, and this comes from samples like tumor samples. So these are processed to extract proteins. The proteins initially start out as whole proteins, but they are treated with an enzyme like trypsin, which cuts at particular sites, and that is to break the protein up into smaller bits that can be easily measured by the mass spectrometer. Then these are separated by liquid chromatography into fractions, and that is to simplify the peptide mixtures so you can basically, the mass spec can detect all of the different peptides that you have in your sample, and then fed into the mass spec. In this case, we're talking about a tandem mass spec. This identifies what we call the parent ion, which is the original peptide that you have. It separates it out and feeds it into another chamber where it is itself ionized and broken into pieces. And so those pieces make a signature, like a fingerprint, that we call a mass spectra. And the fingerprint is unique to that particular peptide, and it can be matched with a database. So if we're looking at tumor samples from humans, we match it with a database where we know what the peptide signatures are. And then the final step is to take the individual peptides, which we identified, and to recombine them into a total abundance measure for that protein. And as I said, this is done for tens of thousands hundreds of thousands of peptides and tens of thousands of proteins per sample. So it really generates a lot of data. You might be more familiar with transcriptomics, which is the measurement of RNA. And that can be done using sequencing technologies and has really become a commodity. So it's relatively inexpensive to get transcriptomics done. You can send it away to a company and you can get high quality results very quickly. For proteomics, that's not such a commodity. It is more that you have to know a research center that does this or a core facility. The cost and the time is much more expensive. So you kind of have to ask yourself, why are we so interested in measuring proteins? Oftentimes, transcriptomics is reasonably presented as a proxy for protein abundance. So if you remember your central dogma, you have genes coded in DNA. They are transcribed to give RNAs, which then are translated to give proteins. And so since transcripts are so relatively easy to measure, transcripts are often used as a proxy for the abundance and the activity of proteins. And we wanted to examine how true this was. So that in this same paper on ovarian cancer, we looked across all of our 200 patients, tumors, and we matched transcriptomic data with proteomic data. And so now we have the measurement of all the proteins, large number of proteins, and a large number of transcripts. And we matched those up. So we found the cognate transcript for each protein. That is the transcript that's actually coding for that protein. And we basically did a correlation across all of the 200 tumors. So that correlation measure would be 1.0 if every time the mRNA abundance level went up, proteins levels also went up. And every time mRNA levels went down, protein levels also went down. You would get a very high correlation. And if there's no relation between these two profiles of the mRNA and protein, you get something that was right in the middle, so a 0, which would mean kind of no association. And the surprising thing that we saw is we do see a positive correlation. The median is about 0.45, but it's not very high. You'd kind of think that it would be higher given that transcripts code for proteins, which are then made. So we were really interested in the reasons why this might be. And we looked at functional enrichment at either end. So things that were very highly correlated, or things that, proteins that were not well correlated with their cognate transcripts. So the things that were not well correlated, we found were more composed of large complexes that were kind of doing the work in the cells: ribosomes, mRNA splicing, complement coagulation. And the ones that were highly coordinated were things like interferon response genes and metabolic pathways. And our conclusion from this, or our hypothesis that we derived from this, was that the highly correlated functions are things that are activated quickly, especially in the case of interferon response genes. These could be activated very quickly. We postulate that the mRNA might come up quickly, proteins get made, and then they're basically done being used, and they're all degraded at the same time. And so you get this high correlation because when there's transcript, there is protein. For the other end, with these large complexes, what we think is happening is that transcripts are getting made, the protein complexes are getting made, and then the protein complexes go and do their work, and they hang around for a long time, and the transcripts get degraded. And so you get a lack of correlation, or even a negative correlation. And so what we really took from this is that protein measurement of proteins is useful. It can give you information that you can't obtain from looking at the transcript. And this has been replicated in across a number of different tumor types now, and by multiple different groups looking at different systems, with this kind of moderately correlated but not well correlated behavior of transcript versus protein. So I'm going to shift gears a little bit and talk about this identification of this signature. So ovarian cancer is a, if it's caught early, there's a fairly good survival rate, 5-year survival rate. The problem is that many ovarian cancer tumors are caught in, not caught until stages 3 or 4, and then the survival rate goes down drastically. So it's an important problem. It's a deadly disease. It's also a disease of chromosomal instability. So a lot of mutations and a lot of chromosomal rearrangements. These can be captured as copy number variations or aberrations. And that means that regions of the genome that are duplicated or deleted to cause differences in kind of the genomic structure of the cancer. So we had, from these 200 tumor samples, we had multiple sources of data. We had genomic data about the rearrangements and the mutations. We had epigenomic data that was epigenetic data that was on the methylome. We had transcriptomics, we had proteomics, and we had phosphoproteomics. And they were all matched for the same tumors. And so this provided a rich source of information to develop signatures. Our first effort was to see if we could predict survival time. And so if we had these tumors that were taken at the time of diagnosis, the samples were taken at the time of diagnosis, we have this rich molecular multi-omic data. What can we do with that to be able to build a predictor for survival time? So at the top, we take the omics data as in kind of as a whole. So multi-omics data stacked on top of each other. We map this to pathways, and that's to provide a functional aspect to this predictive method. And then we use machine learning to train a model based on the survival time of the patients from this retrospective study. And we did cross-validation, which is a way of internally validating that we've got a good fit for our model. And then we also did a separate evaluation set, which I'll talk about in a couple of slides. This was very successful. So the outcome of this paper really was a signature, and I'm so glad that Kaplan-Meier plots were shown in the last talk. So I'm going to briefly go over this. This is the time in years on the x-axis. On the y-axis is the probability or the overall survival percentage. In blue is our signature. This is about 90 proteins that we identified. And it is when this is relatively up in tumors, there's a much better survival than the bottom line, which is in red, that if the signature is relatively down. So if the abundance of these, the signature is high, you have much better survival, and if it's low, you have much worse survival. And so that was kind of the outcome of this study. And it was really interesting because I was talking with one of the collaborators on this study who is a clinical researcher, and he said, yeah, this is a great demonstration. I think we've really nicely shown that we can use omics data to do something, you know, kind of interesting, but I don't find it all that useful. And I said, what, really? And he said, yeah, because I see patients, and if a patient comes to me and I diagnose them with this deadly disease, it doesn't do me much good to be able to predict how long they're going to survive if I can't do anything about it. And there are very few tools for ovarian cancer to be able to do something different than the existing treatments. And so that was really, for me, really stuck with me. And it's, I think, an important point, and it kind of leads us to our second portion, which is about determining the function and trying to get at mechanisms that could be either suggest alternate therapies or be used to design alternate therapies. So I'm not going to spend very much time on this. We used the same kind of approach, which is a statistical enrichment approach, but now we're using a combination of data. On the y-axis are different pathways that we've identified using this model. And these columns are transcriptional data, proteomic data, phosphoproteomic data, and a combination of all three. And basically what from this, we were trying to discriminate between long surviving ovarian cancer patients and short surviving ovarian cancer patients. And you can see that these, that the combination of different multi-omics really gives a much clearer picture of what's going on. So the final portion of this story is that we then took this original study and we performed a prospective study. And this was gathering tumors from existing patients and analyzing them using the same stack of data, obviously for each of the tumors that we got. And so one of the things that we did was we initially validated that signature that I showed you. Unfortunately, because of the way the study was designed, we were not following the patients for their survival time. So we really couldn't fully validate that, but we got some pretty good indications that that signature held in a prospective study. And then we used pathway analysis and multi-omics, and this is a kind of a cartoon version of our final model where DNA damage flows through this signaling pathway, which we were able to get by phosphoproteomics, and affects the cell cycle replication. And this is really a nice way of kind of tying these things together. Now, this hasn't been used for translational. We're not actually making any differences in how patients are treated with this particular study. But a follow-on project, we're collaborating with a group that is looking at acute myeloid leukemia in patients and running clinical trials on different drug treatments. And so we're actually building models to be able to predict which drugs might be most effective for which patients using a very similar approach. So that's kind of where we're going in the future. So I'm just going to wrap up with a cautionary note. This is all fishing in big data and using machine learning comes with some caveats. And some of the caveats are that you can almost always catch something if you're fishing in a pond this big. It may not always be what you want to catch. So correlation is not causation. And this is a slide that I like to show because it's a great, the website's on the bottom. It's a guy who's gone through United States, publicly available United States data and found these trends that provide very good correlation. So on the top, for instance, he's correlated divorce rate in Maine, the state of Maine, with US per capita consumption of margarine between the years 2000 and 2009, and he gets an R value of 0.99. Obviously, these things are completely unrelated, but it's funny that you can find correlations like this. So I'd like to thank you for being an attentive audience. I'm hoping that I did not bore you too much, and I would be happy to answer any questions. I'd also like to acknowledge the team. This is the core team that that is working on the cancer project. There are many more people involved than this. And then our NIH, NCI, National Cancer Institute program that is aimed at characterization of tumors, proteomic tumors. I would also like to thank.
Thank you, Dr. Jason McDermott. That was a wonderful talk on how omics could be used for mass data analysis generated from high throughput sequencing data as well as the proteomics analysis, so that we can arrive at a meaningful conclusion, segregate the data, and then we have an understanding of what's going on. Do we have any questions to the speaker?
I was crystal clear.
Yeah, just wanted to ask, omics, can we really, is there a situation where the proteomic data and the genomic data or the transcriptomic data do not correlate? And also there are multiple variables. So how do you remove the, variables that affect the data?
Yeah, that's, I almost had I have a slide that goes at the end of that correlation is not causation. And that's a really important point. It can be very difficult to account for all the potential confounding variables. There are statistical ways that you can look to see if there seem to be confounding variables. And so we use some of those. Sometimes I think it's oftentimes, especially in studies like this, metadata, clinical metadata is variable itself, right? So it's been collected by different people, sometimes using different systems. The nice part about the study that I talked about is that one of the things that they really aimed at doing, this is based on the Cancer Genome Atlas project that was funded by the NIH for a long time, is they really tried to be very careful about collecting the same kinds of metadata. Even then, it is, there are statistical models that will basically allow you to take that metadata and see if there are influences from age or from, treatment regimen or, from other factors. And we didn't find any of those in the signature that I talked about. But you're never quite sure if there are other things outside that that weren't captured, right? And so I think that's why it's important, and that's why I like to talk about this study, because the program that funded us had the foresight to say, oh, we've done a retrospective study of all of this data, and it's very easy to come to conclusions and good predictions because you can work with the data over and over and over again, right? And then we did the prospective study to validate these findings, and I think that was really important. And they did that with a number of different tumor types.
Thank you, Dr. Jason. Dr. Jackson, do we have questions?
No, I don't see anyone else, but I would agree with you about the correlation, because many times we get carried away with looking at the correlation and seeing that it's something that we would be expecting, or yeah. But what
Yeah.
are your views about using the database of single cell sequencing that is just coming up now? Like
Oh,
that would give you much better, because you have a lot of replicates in that, right?
yeah, yeah. So single cell, single cell information. So transcriptomics and genomic data is really an interesting area, and I think it's going to have a lot of applications. Interestingly, it is now possible to do single cell proteomics, although it's again much more expensive and slower. But that's going to be a very interesting too. So all those correlations, when I was talking about the correlation between mRNA and protein, we're looking at, hundreds of thousands or millions of cells all mashed together. And so they are not synchronized. There's lots of different things going on. But once we can get to the single cell level, it'll be very interesting to look at that correlation and see if what kinds of things we can get from, these kinds of multi-omics measurements that we might be able to eventually do on a single cell. We might be able to get multiple types of data from the single cell. And I think that's really exciting. In terms of what can be done right now, I think the transcriptomic data is really interesting. So the single cell transcriptomic data, and it's really showing like the heterogeneity of these tumors. That's another thing that we didn't, we were working with bulk tumor samples, which means that we took a bunch of a chunk of the tumor that is probably fairly heterogeneous in all of those modalities and mashed it all together. And the single cell analysis is really showing that, of course, that there's lots and lots of stuff that's going on. There's lots of variability at the individual cell level. So I think that's really fascinating too.
Thank you, Dr. Jason, for a very interesting talk on integrating omics to understand complex biological systems. I also thank you on behalf of all the organizers, and I think the talk was very useful to not only to us, but also to all the young students who are listening to you. Thank you.
Thanks for the invitation.
Thank you, Dr. Jason.